You've had an overview of our process. You know how to identify interventions. You know how to describe the study designs. And now we're going to get to the final step of actually writing up the paragraph, getting to the evidence, and putting it in its final form for our purposes. So, what should a summary do? Well, we know that it should summarize, because if it doesn't, it's not serving its readers at all. You might as well direct them to the original paper if you're not being brief. So we don't want to report all of the results, we just want to report the relevant results. You should try to do this in about 150 words, but there are some exceptions. Primarily, the paragraph should be trying to answer the question, what was the intervention, including the methods, and did the intervention work, including the data? And this is a really important part of our process. We include the data. So if you look at any of our paragraphs and it tells you that mowing increased the number of native plant species, it'll then tell you how many species there were found in the mowed treatment and in the control treatment. And so from that, you can then assess for yourself whether you think that was a strong enough intervention to merit you trying it on your own reserve. So that's a sort of measure of an effect size. If you manage to do that, and you still have some words left over, or you think it might be really important for the reader, you can then try to answer a few other questions, like, how did the intervention work? Not did it work. So did it work might be saying, what was the effect in the treatment versus the control? But how did the intervention work might be comparing two different kinds of treatments, saying, okay, well, we tried mowing twice over the summer, or we tried only mowing once. Different flavors of the control or of the treatment are sometimes tested in the same paper. That's interesting information, but we don't need to know it until we know, does the intervention work? So try to communicate that first, and if you've got space left over, turn to these details. What else happened? Sometimes they'll report a lot of things that are not about the intervention per se, but they might really help the reader interpret the effect, or whether it would be relevant to their study system. So that's all interesting stuff, but again, remember, we're trying to summarize, we're trying to be brief, and we're trying to answer those two questions. What was the intervention, and did it work? If you do that, then you can move on to other stuff as gravy. So, here is a template for how we write paragraphs. Here's a paragraph here, color-coded, and here's, it's very formulaic. And we do it like this for a reason. It's not the most exciting thing to read, but once you get used to reading these paragraphs, you know exactly what format they're going to have, and you know exactly where to go for the information. So, at the beginning of the paragraph, we're always telling the readers what kind of study it was, and this is something we've been practicing throughout the day. So here you've got a replicated controlled study. What was the year? In 2000. How many sites? What kind of habitat? Well, they're just saying of bottomland hardwood forest, so they've missed out to how many sites. In region and country, in Georgia, USA, etc. Every paragraph starts with this format. So we start with that study design that we've been looking at throughout the day. And then we move on to the main results and the intervention. So what was the intervention here? It's color-coded in blue, burning and unburning. So the intervention was burning, and they compared burned and unburned stands. And what was the effect? Abundance, diversity, and richness were similar. So there was actually no difference in this case. By reading that first sentence, you should get the main message from that study. You should know, did the intervention work? You should know what kind of study it was, and you should know where it was done. Down here, then, we might give you some additional results, and we might then incorporate the data. So here, I was saying we actually have a measure of effect size in these paragraphs. You can see in the burned sites, there were 43 amphibians, that's abundance, yeah, and there were 62 at the unburned sites. So from that, you can decide whether this is an intervention that would make enough difference that you think it's something that you might want to implement in your reserve. There's a difference between something that makes a big biological difference and something that makes a big statistical difference. And throughout this process, we are only reporting statistically significant effects. So if you read in the paragraph that there was an effect, you can assume it was a statistically significant effect. But that doesn't necessarily mean that the effect is big enough that you'll care about it. And so that's why we always include these numbers so that the readers can make that decision for themselves. I won't go over this again, but you saw this earlier today. This is what would go in that first sentence. Yeah. So this is very important. This is a very summarized way of reporting data. We often report So here's the treatment, intercropped cabbage, and the control is cabbage alone. And then throughout this, we're reporting the data in that order. So we're giving you 16 versus 52, 110 versus 305. That's always in the order treatment versus control. And we try to maintain that order throughout. So it's this data versus data format. Right. If you've got a replicated study, as you do here, one of the main things we want you to report in the methods is how many replicates were there. This is something that can be difficult to assess, and I think this is something we'll talk about later. Sometimes someone will take many samples of a single intervention. You might have a single meadow where you mowed the grass, and then you have multiple quadrats in that meadow. Or you might have multiple meadows that you mowed, and you might have a single quadrat at each of those meadows where you're measuring plant diversity. Those represent different kinds of replication, and it's a bit tricky to figure out what you should report here. So we'll talk about this as you go through your examples later. But just be aware that you should be trying to report the level of replication if it's a replicated study. So there can be a lot of data in one of these papers, a lot. And you need to filter through it and figure out what, if someone else from your team were summarizing this paper, what would they pull out? You want the sense that it's going to be standardized from person to person. And I think it can help to standardize if you, at the beginning of the process, come up with a list of things that you think you're going to report. This is rather new. Bill mentioned at the beginning of the day that our process is evolving, and something that I highly recommend now to new authors is when you talk to your advisory board, agree with them a list of metrics that you're always going to report when you find it in the paper. So this, from the Mediterranean farmland synopsis here, we were looking at multiple ecosystem services, and there's so many different ways of measuring these ecosystem services. So soil, for example, soil can be so many different things. You could be looking at the organic matter in the soil, you could be looking at erosion. And every time one of those metrics came up in a paper, I wanted to know that whether I should report it or not. And so we sat down with the advisory board and said, what are the things that you think people really care about? Whenever we find that metric, we're going to report it. So I think this is a great way of deciding and standardizing the kind of information that you're going to put into that summary. And sometimes there will be a trade-off between getting your 150 words and getting all of this information into the paper. So what you might also do with your advisory board is create a list of priorities and saying, we're interested in all these metrics, but we're only going to report the highest priority if there are too many to report, really. Yeah, be systematic. I mean, I would hate to think that there's a paper out there that reported something that we were interested in, and one person writing up a summary would report the effect and another person wouldn't. I'm sure that has happened. I'm sure it will happen. But the more systematic we can be, the better, more defensible it is. It's really important to report what doesn't work. So we call our flagship publication What Works in Conservation, but we also want to know what doesn't work in conservation. We don't want people wasting their money and their time on things that don't work. And especially if you're being systematic, I think it's really important to report things that don't work. Because at the end of this exercise, we're going to end up with a bunch of summary paragraphs that are all looking at the same intervention. And if we only report the ones that work and we forget to report about when it doesn't work, we're going to get an overwhelming picture that this intervention actually does work. So we actually want to be able to, at the end of the process, to say we looked at 10 different studies, 5 of them it worked and 5 of them it didn't. So just because something doesn't work doesn't mean you should not report it. You very much should. And of course, as I mentioned earlier, if you're reporting what does work, it should be a statistically significant difference. Do not report the results if they were not tested statistically. So if for example, the author says, okay, we mowed these 2 grasslands, we counted the number of native species in each one, there were 10 in the mowed site and 8 in the unmowed site, and they leave it there, we don't comment on that at all. We don't say there were more, even though there were 10 species in the mode treatment there. They haven't tested it statistically. We don't actually know if 10 is actually different from 8. That's a weird concept, it's a statistical concept, but if they haven't tested it, we don't comment on whether 10 is more than 8 or 10 is equal to 8 or whatever. If it's been statistically tested, then we'll comment on it. So if they say that actually there was not a statistically significant difference, then we would say, okay, mowing didn't have an effect on native species, 10 versus 8 species. Sorry?
But we still report it, right? We still retain those studies.
Potentially not. So if they haven't tested it statistically, you probably wouldn't report that result at all. But if they've tested it statistically and there's not a significant difference, then you report it for sure, definitely. Mm-hmm. So that's this point here, report what does not work. If they haven't tested it at all, we probably wouldn't report that. That doesn't mean that there might not be something else in that paper that you would report. So another challenge that we often have is that studies are very complex. So they don't just go out there and do this hypothetical mowing that I've been talking about all day and just measure the number of species. They'll do things like, okay, we mowed each of these 2 meadows, and then we burned half of them, and then we came back and counted the number of species a year later, and then we came back and counted the number of species 2 years later, and they give you these huge tables of results, and you don't really know what is the overall effect. So one thing you really have to do is you have to learn how to filter. You have to learn what is the data that I'm going to report. And I think the key question to ask yourself is, what is the overall effect? If you had to just tell someone, does mowing work? You might not actually care about whether it was mowed and burnt or mowed and measured 2 years later or 1 year later. You might want to know overall, does mowing work? And so you'd probably be looking for some way of communicating the overall effect of mowing. And I think you should think about that when you're looking for the data points that you can potentially extract from that study. Another problem is that multiple taxa will be reported. So beetles, for example. Let's say you're doing a synopsis on beetles, and they report the effects of mowing on ground beetles and ladybirds, and you're not really interested in that. It's too much information for a synopsis. You're really interested in, does this work for beetles in general? Always be thinking about, how can I report that overall effect? If you've got extra space, then by all means, include that extra detail. But always remember that we're trying to summarize. If we provide too much information, we might as well say to them, go and read the original publication. We're not summarizing, we're not really doing our job. Here's a paragraph in which they have given an overall effect, but then they've gone by taxon here, by species, and they've broken it down. So if you've got space, by all means do it. But here we see that the total number of ground beetles is greater. This is really challenging, and this is something that we're going to look at in some examples later. One strategy we've come up with for dealing with this is using this phrase here, sort of in X of Y comparisons. So here's an example of a study that was on cover cropping compared to having a bare fallow. And we can see that in 2 of 4 comparisons, it helped broccoli yields. So higher broccoli yields were found in plots with winter cover crops compared to bare fallows in 2 of 4 comparisons. That's giving you the overall picture without wading into the details yet. An attempt has been made here to then give you some more information about what those 2 of 4 comparisons were. They were with Phacelia as a cover crop. And when you read into the methods here, you get a sense of what some of the complexity of this study design was. That half of the plots had Phacelia, half had Mercet rye, and that there were multiple different crops. And so actually in this case, the 4 comparisons here represent 2 complete different cropping seasons. But you don't need to go into that level of detail at first. When you read this paragraph here, it just tells you 2 out of 4 times growing cover crops helped crop yield. And I think that's what we're aiming for here. We're aiming for the overall picture, did it work, and then if there's space, you go into some of the detail. By all means, we want to report as much of this methodological detail as we can so that people can decide if it's relevant to their system or not. But we're actually doing a disservice if we're focusing on too many details and not giving them an overall effect. So this is a good thing to bear in mind, this phrase here in X of Y comparisons. And often then you have to figure out how you're going to report the data, and it often works very well to just have a range. So these might be the mean values for those 2 crops that had higher yields, and these would be the mean values for the 2 crops that had lower yields in the cover crops, in the bare plots. And we just report it as a range. So rather than trying to have any derived statistics, we actually just look at those 2 values, we just say, here's the range. So we haven't computed anything here. We've just given a range of values. So here's an example of where you might see this kind of complexity. What we're looking at here is a study on weeds. So we're trying to control weeds in farmland, and we're trying to do that with cover crops. So it's just like the last paragraph we're looking at with cover crops versus without cover crops. But they've measured weeds 50 days after transplanting. They've done that in 2011 and 2012. They've also measured weeds at pepper harvesting in 2011 and 2012. And they've measured weeds in two different ways. They've given us weed density, and they've given us weed above ground biomass. So what's the overall effect here? If you grow cover crops, what happens to weeds? We need to take this kind of complexity, because they haven't given us an overall figure here. They haven't said in general, over 2011 and 2012, 50 days after transplanting and at pepper harvesting, what happens to weeds? That's now our job. So we've tried to come up with a sort of standardized way of dealing with something like this. Also, I forgot to mention there's two different cover crops here as well. It's not just a single cover crop. You've got oat and you've got hairy vetch, and here's the control. So again, in terms of study design that we were talking about earlier today, you've got your treatment and your control. These letters here you'll often see, again, I don't want to go into the statistics too much, but it sounds like people have heard about statistics. They're a bit familiar with them. Basically, two of these entries here are statistically significantly different if they don't share a lowercase letter in common. So here, in this case here, the no cover crop plot, this 58 here is statistically significantly different from the 24 because this has the letter A and that has the letter B. They're not both letter B. So what we tried to do to summarize this sort of paragraph is use this X of Y comparisons notation. So the sentence we come up with here at the top is more weed biomass was found in plots with cover crops compared to plots without cover crops in 6 of 8 comparisons. This is not even the most highly summarized form because this is just talking about weed biomass. So we're just looking at this section of this plot here. But in 6 of 8 comparisons, so how did we get to that? You see in the parentheses there's a couple of different figures summarized there. There's the 50 days after transplanting. So let's look at 50 days after transplanting. This one here, you'll note, is not statistically significantly different. There's a lowercase a there and a lowercase a there. But this cover crop is different from the control. This cover crop is different from the control. This cover crop is different from the control. That one's not. And we've got 1, 2, 3, 4, 5, 6, 7, 8 comparisons there. So in 6 of those 8 comparisons, there is a statistically significant difference between some kind of cover crop, whether it was oat or hairy vetch, and the control. And we've then tried to give the ranges of those values. So if we look here, that's 50 days after transplanting, 16 to 25 versus 37 to 58. So if we look at, remember the data is always presented in the format treatment versus control. So the 16 to 25 refers to the data for the cover crop plot. So if we look here, we should see, okay, 16. We generally try to round things off if we can to make it a bit simpler. 16 to, well, it would have been 32, except this was not a statistically significant difference there. So we've gone for this one here. The range of values we presented there is 16 to 25 versus 37 to 58. So that's here, 37 to 58. And we do the same thing then for pepper harvesting over here, and I haven't illustrated that. And that information has just gone into the parentheses, but the main takeaway message there is that if you're interested in what happens to weed biomass, 6 of 8 times, there's going to be a statistically significant difference. And I think that's the level of simplicity we need to get to make these things useful. And then if you can, you communicate some of the nuances there in the parentheses or in the methods or what have you, but someone reading this paragraph will get a sense of whether it works and how often it works. So that's messy, right? That's messy, but it's not as bad as it gets because they've given us the numbers, they've given us the statistical comparisons, but sometimes they don't. Sometimes they just give us a figure, and we then try to extract the data from the figures. And we only do that if it's not available in the text or in tables. There's one program that I use that's really quite good, and I might just give you a quick demonstration of how it works if I can. Web plot digitizer. When I first heard about extracting plots from graphs, I was skeptical that this was a good thing to do. But actually, I found it to be a pretty robust process, and this tool makes it quite easy and quite repeatable. Okay. Because the great thing about this program, normally when you're working with PDF here, you can just you can do a screen grab of one of these figures here, and then you can just paste it in here. And what's great about this program is that it allows you to align your axes here. So you can see we've got this axis here is going from 0 to 100. And you can do this even in some cases too. You can figure out what the standard error is on some of these bars if you need to sort of like, after the fact, figure out if there was a statistically significant difference or not. So we actually make quite a bit of use of this web plot digitizer because data reporting is really pretty shockingly bad. And you can get a pretty robust estimate using this tool here. So I just wanted you to be aware of that and to see how relatively easy it is to use. Okay, so what do you do if there are multiple interventions in one publication? So you were looking through those abstracts earlier today, and you said, okay, well, here's the intervention. But what if there are actually multiple interventions? What if not only did they mow, but they also burned, and you're interested in mowing and burning? What you'd want to do there is you'd want to write a separate paragraph for each intervention. Because ultimately we're going to try to get all of the paragraphs on mowing, and we're going to get all the paragraphs on burning, and we're going to put them in separate places, and we're going to extract the key messages on those. And it's okay if that means that a lot of those paragraphs are identical. So, we've talked about the study design. It's quite likely that your first sentence for both of those paragraphs is going to start in exactly the same way. It might not, though. You got to be careful because the study design might be slightly different for the different intervention. An sort of inverse problem is what happens if you have multiple publications that are actually writing up the same experiment. This often happens. You'll have a single lab group. They'll do one big field trial, and they'll have multiple students who are writing up potentially different aspects of it, but sometimes literally the same thing. And that's sort of a problem because when we then go to try to say, okay, 5 out of 10 studies found a positive effect, it might actually be that it's more like 4 out of 8 or something like that. Our view is that we should summarize all of those studies anyway. You probably end up having to practically summarize before you realize it's the same study. And be careful because sometimes it's not. Sometimes it superficially looks like the same study and it's actually it's a different experiment. But if you find something that's literally something you've written up before, we usually just say same study as. So this is the reference here. So this is referring to some other paper, and it's the exact same as that one there. So then people can look later and say, okay, well, when I'm taking account of the effectiveness here, I should bear in mind that there's not as many replicates as there might have been. There's not as many different studies. And this is useful because often there will be areas of overlap, but then there will be bits of new data as well. And that's why I think they feel justified in reporting, in publishing multiple papers from the same experiment. They'll report the same information on crop yields, but then someone will report information on birds, and someone will report information on soils. So often there's some overlapping information, but there's also some new information. And this allows us just to link between those studies. All right, so sentence structure. I mentioned this a little bit before, but I think this is really important. When you're writing a sentence, I think it's always important to mention the treatment first and the control second, and then present the treatment data and then the control data. That's up here. And the reason this is important is because if you do it this way, then your words mean the same thing throughout all of the paragraphs you're writing. So when you go to look at all of these paragraphs that are on the same thing, and you see the word fewer, you always know it's going to be fewer in the treatment, not fewer in the control. If you start switching these word orders around, you end up with this sort of situation here. So more bird species were found in restored habitats compared to unrestored habitats. If you flip around that word order, you could say fewer bird species were found in unrestored habitats compared to restored habitats. This means the same thing. But when you're going through and reading 20 paragraphs on this intervention, and you see more and fewer, it's very confusing if they mean the same thing. So I think it's far more useful if more always means the same thing, and it will always mean the same thing if you write in this order. So write treatment versus control, treatment data versus control data, and when someone's quickly reading through, they'll know that more is a good thing for birds. One other really important point, though, is I think you should always be clear about what was compared to what. So, it's not that useful to know that more bees were found in plots with cover crops. Compared to what? Compared to plots with other crops? Compared to bare soil? Compared to burned plots? There's so many different comparisons, and you might end up having different studies on the same intervention that have different controls. So I think you should always be clear about what the control was for this paragraph that you've written up. And so we use this phrase, compared to. More bees were found in plots with cover crops compared to plots with bare soil. And I think you should try to refer to this as something other than the control. We know it's the control. Describing it as the control is not particularly informative. So I would try to try to avoid using that phrase. You're going to have you will have described the study as a controlled study in the first sentence already. So there will be different kinds of controls in different paragraphs. Right, so once you've answered that main question about does the intervention work, when you look at the treatment versus control, does the intervention work? You might have these other questions about how does it work. You might have comparisons of treatment versus treatment instead of treatment versus control. So an example here would be more eggs were found in nest box X than in nest box Y. This is not testing whether nest boxes work. We're not looking at the nest box versus a control, which is some absence of a nest box. But we're looking at nest box design X versus nest box design Y. So really this is a treatment versus a treatment. This is what we've been calling implementation options. This tells you about how you might install a nest box, not about whether installing a nest box would work. It's great information, but it should be a secondary category of information. It should be a secondary priority when you're writing up your paragraph. If you have enough studies that have looked at this, though, this treatment versus treatment, you might just treat that as a different intervention. So it's no longer about asking whether nest boxes work, it's about asking whether nest box X works better than nest box Y. It's still a conservation intervention, but it's got a different kind of control. And so the rule we have in place is that if there are 5 or more studies that make this same comparison, then you don't report them as implementation options. You create a new intervention that is the intervention would be use nest box X, and you summarize them for that new intervention. So always be asking yourself when you're about to put something in your paragraph, am I actually comparing a treatment versus a control, or am I actually comparing a treatment versus a treatment? We always try to write in plain English. We know that we're not necessarily writing for a scientifically literate audience, and so we try to simplify wherever we can. So here's a bad example. Riparian restoration resulted in an increase in the populations of Aphidophagous surfidae. And here's a simple example. More aphid-eating hoverflies were found on restored riverbanks. There are so many different forms of jargon out there that I can't possibly go through them all, but just try to keep an eye out for terms like riparian restoration or surfidae or Aphidophagous and explain it in a simpler way. We have some recorded talks on writing in plain English. And there's other house styles which are included in a document that you'll be given later, like we always use the Z spellings instead of the S. And there's house style on numbers. Nancy showed you earlier that we've got this template that is useful to fill in while you're writing your paragraph. These are IUCN categories for basically, well, in this sense, action is a broad version of an intervention. So one action might be vegetation management or even mowing or something like that. But this is a sort of generalized version of that intervention. Our database is tagged with these so that when you go to the Conservation Evidence website, you can then search by action or threat or habitat. So you should write those things down as you're going. Likewise, the taxon. You should also download the RAS file while you're going, because that's something else you'll need to upload later. And we're going to take you through all of this later. So this is going to be your first effort to actually try to do some writing. We've got here a badly written paragraph. It's not that badly written, but it's kind of badly written. And it's on that vineyard study that we were looking at earlier today. So what I'd like you all to do is read this, look at the study, and see what things you think are wrong with this, and maybe actually try to rewrite it. So you're going to need to look at the original study. You might need to extract some data from graphs. You can eyeball it. You don't have to go to Plot Digitizer today. And you should have a copy of the guidance. But how did that go? How did people find that? Initial thoughts. So that's something I want to talk about here. What kind of study design would you call this?
Well, I'd say it's a replicated control study.
Okay. So you call it replicated.
Yeah, but I didn't know that until I got into the text.
Are there takers on that? Do you think it's replicated or replicated? So that's one thing I was hoping people would pick up on when they were looking at that exercise earlier today, is that when you read the abstract, they say, we did this to one plot and we did it to another plot. So you think actually it's not a replicated study. Arguably it's not in some ways, because they've actually only applied the treatment once here and once there, and they've taken a bunch of what could be called subsamples. So it kind of depends on what you think the actual experimental
exactly
data
the same
is.
thing.
And right, exactly. And so but even within, say, the Merlot treatments,
Yeah.
even within that one scattering of seeds, you could argue that actually the arthropods here are different from the arthropods
Yeah.
here. So sampling them here and here is actually, those are independent data points, and we should consider those to be replicates. But this is the sort of issue you sometimes need to wrestle with, and that authors sometimes have not grappled with very well themselves. So I don't think they've explained this very well in the abstract, as you rightly pointed out. But also there are some philosophical challenges there. Do we consider that to be replicated or not? They have just seeded those plots once, presumably. It is just one seeding. So we don't get as much information as if they had another vineyard, but they had also sowed it there, and there'd be other conditions that would differ. And then that's one reason that replication is important, right? Is we start to build up a similar picture at many different sites, and we can infer that any effect we see is probably not due to some background condition that we haven't measured. I think I would call this replicated, and most agricultural studies will use this sort of design and will call themselves replicated. It depends on what's being measured. So if you're measuring something about the water table, I would probably consider it not to be replicated, because there's probably one source of groundwater, say, underneath that whole vineyard. But what you're measuring is something small that's probably encountering the landscape on a very fine spatial scale. Those different areas of that single 0.1 hectare plot probably constitute fair replicates. But these are the questions that we have to wrestle with, and I'm not sure there's ever a perfect answer. I do think this is a debatable case here, so that's good job for bringing it up. I think that's an important point. Other thoughts, things that people wrestled with.
Do we consider this as two separate experiments? If seedling increase plant cover and if plant cover increase arthropods?
If
If seedling increase arthropods.
Yeah.
But first they also evaluate if seedling will increase or not
Right.
plant cover and plant diversity.
Okay, so how would you handle that? How would you replicate
With
that?
two different topics. One is if seedling increase or not plant cover, and the second if seedling increase or not arthropod diversity and species richness.
I think that's probably right in terms of what the mechanism is, but in my mind, there's really only one intervention. All they've done is they've planted the seeds. So from the point of view of the intervention, there's only really one paragraph to write up, I would say, there. And if we get into the detail of whether it was the plants that are increasing the arthropods, I think that's getting into this issue of implementation options, the how it works. Whereas what we really want to know is, does it work? If you take this action, if you plant seeds, if you're interested in arthropods, what happens? And we leave the mechanism in the background. I think we don't speculate on that too much. As scientists, we would love to know that. And I think we would love to be able to say it's not because they seeded it, it's not because they didn't use herbicides, it's because there were plants there. But as practitioners, we just need to know, did it work? And so I would say from the point of view of conservation evidence, we probably wouldn't write 2 paragraphs. But if we were trying to infer something about causality, we probably would want to do a different experiment, but ultimately we would want to connect it to plants or lack of herbicide or whatever. Other thoughts? When looking at this paragraph, were there things that stood out as being obviously wrong? We've already talked about the study design. Obviously, they got the study design wrong here. It's not just a study. We've decided it's a replicated controlled study, so that's one thing there. Country and location, yeah. Yeah, so it says it's Israel here, but it's kind of in the wrong place, isn't it? So if you look at our template, this is not at all the order that the first sentence goes in. So it should be reorganized. Yeah, what do you think about a grumosol soil?
It's too much information for your opinion.
I think it's too much information as well. So I think this is a case of we needed to filter there. That might be interesting to a soil scientist. There were soil results in here, and if we were writing a soil synopsis, that might be relevant. But if for arthropods, it might be too much information. Reference is missing. Very good. Yeah.
With the years, 2013 to 2015 in the paper.
Yeah. Okay. And were all of the experiments done in all of the years? No, because I think the Merlot was only seeded in the second year or something like that, right? So you got to be careful what things you're reporting. The authors might tell you that they did the study at one time, but actually the results that you're reporting are just a subsection of what was reported in the whole paper. What about this here? These results here?
No number. No.
Exactly. So that we never report the statistics. We never report P as this. We want to know what were the numbers in the treatment and what were the numbers in the control. It's important that it be significant if we're reporting it as a difference, but we never want to communicate that. We want to know the actual numbers of the arthropods. Does anyone know what an arthropod is? Is that an acceptable word?
Insects.
Insects? Yeah, potentially. So,
It depends if they are only insects
or whether they're arthropods or not. Yeah.
spider.
Sometimes it's challenging, and sometimes it is insects and spiders, or insects and mites, or something like that, and sometimes you're just stuck with these terms. In this situation, I think we might have to just live with arthropod. But yeah, if you could say insect, I think that would be nicer.
Is it post-trimming an intervention because it was caused by the drought? And that's when they decided it's G2.
point. So would you call this a before and after study?
Well, just suggest
So I think
that they might.
I think
halfway through the year, doesn't
it is interesting, but I think when we decide what the study design is, we always need to think about how the data is reported. And they haven't, I don't think in this case, if you look at the figures and the data that's reported here, so they haven't given us a statistical comparison between the pre-trim and the post-trim. They've only given us a comparison within here. So we can compare the treatment and control within pre-trim. We can compare the treatment and control in post-trim, but we can't actually compare the pre and the post the way they've given us the results. And so for that reason, although this could have been a before and after study, and I was hoping someone would pick up on that, I don't think we would call it that because of the way they've reported the results. They haven't actually used that. I mean, so that would be fantastic. We would have a lot more confidence in the results if they had presented it in that way. But I think you always need to look at how the results have been reported. Have they actually done that comparison? I think one point here that could be made is that if we want the overall message of did this intervention work of seeding, we might not want to know whether it just worked pre-trim or whether it just worked post-trim. That might be too much information. So I think this might be an example of where you might want to use that in X of Y comparisons notation. You might say overall, if we looked at the pre-trimming and the post-trimming, how many times did this work? So I think that might be another way of handling this level of complexity here. This might be too much complexity for someone to get at on their first reading. They were classified to order level except for attacks of special interest such as parasitoid wasps. Yeah, that's kind of a bit random, isn't it? Why have that in there? Good. Okay, good. Well, you can see how challenging this is in the real world, and if this were going to go into conservation evidence, there's a good chance that it would be in a synopsis on just arthropods or just soil or just something. So you probably wouldn't be faced with digesting all of these results on so many different groups, but it shows you, the challenge of real world studies. And someone else might be writing that synopsis. So if you're as an author, if you're going through that journal and you're looking for studies, and it's you're doing a synopsis on birds, you might see this, and you'd need to pull it because other people might be writing that synopsis on arthropods or soil or crop yield or what have you. So as part of your process, you should check to see if it's been summarized on conservation evidence. And you probably will need to write a new paragraph anyway because your synopsis will be on something that theirs was not on. So there might be a chance that someone could have written this up for a study on Mediterranean. I might have written it up. And in fact, we would have included all of those things. But let's say someone was doing a synopsis just on plants. They might have missed out all the information on arthropods. You probably, even though it was already on conservation evidence, you probably have to write a new paragraph just on arthropods. But yeah, we'd certainly want to search the website first and make sure it hasn't been written up.